All right, so welcome to part 2 of the macromolecule lectures. This component of the lecture series will cover proteins and nucleic acids. So starting with proteins. Proteins are incredibly important. They account for more than 50% of the dry mass of most cells, and they are instrumental in almost everything an organism does. Humans have tens of thousands of different proteins, each one with a specific structure and function. In fact, proteins are considered the most structurally diverse and sophisticated molecules known, and that's because each one has a unique 3D shape or conformation. And as we know with biology, shape and structure influences function. So because of those differences in shape, each protein has a different function or is able to play a different role in the body or cell. So with proteins, proteins are, their monomer subunits are amino acid. So amino acids are the monomers of polypeptides. Polypeptides are the polymers of proteins. So proteins are all polymers or sets of polymers of amino acids. And those sets of amino acids form polypeptides, which are the polymer. Your amino acids are organic molecules that all have this kind of these four groups that associate around an asymmetric carbon. So every amino acid has an asymmetric carbon that is surrounded by a carboxylic acid group or carboxyl functional group, hence the name amino acid. Everyone contains hydrogen, an amino group, the amino component of the name, and then a variant group or an R group. The R group is known also as a side chain, and that's the component that differs with each amino acid. So the diversity within the 22 different types of amino acids, we can actually narrow it down to 20 because there's 20 that we focus on for coding in genetics. The variation between those different amino acids arises from this variation in side groups. Your side groups all have different chemical characters, so they cause the amino acids to behave differently chemically, right? So they're classified as either being nonpolar, polar, or electrically charged based on their side chains. The side chains that are electrically charged can either be acidic or basic. And the reason why that is important because the chemical character of those amino acid side chains are what will influence the folding of the polypeptide chains, which then influences structure, which will influence function. Alrighty, so when two amino acids or more are positioned so that the carboxyl group of one amino acid is adjacent to the amino group of the other, an enzyme can come along and cause them to join together. And that enzyme will catalyze a dehydration reaction to cause or produce a covalent bond that's known as a peptide bond. So amino acids, your monomer subunits of polypeptides, which are your polymer units, are joined together by peptide bonds. And again, that peptide bond is forming between the carboxyl group of one amino acid and the amino group of the other amino acid. So usually at the end of one polypeptide chain, you have a free amino group, and at the opposite end, there's a free carboxyl group. So you have peptide bonds forming or linking these monomers along this kind of backbone unit. This is what creates your peptide backbone. And then radiating off of that, you have your side chains. So again, the linkage of those amino acids by peptide bonds creates your polypeptide, which is the polymer unit for proteins. Now, before I move on, that amino acid sequence that comprises a polypeptide, it's not random. It's actually determined genetically. And we'll get into that more when we talk about nucleotides. But polypeptide, that term is not quite synonymous with the term protein. Right? You think of protein as a sweater. Polypeptide serves as the yarn that makes that sweater. The protein, which is the sweater, is again comprised of polypeptides or different yarn, which creates a sweater of varying shape and size. So my analogy is that a functional protein is not just a polypeptide chain. Rather, it's one or more polypeptide chains that are precisely twisted, folded, and coiled into a specific way to form a molecule that has a unique shape and therefore a particular function. And again, it's that amino acid sequence of a polypeptide that will determine the 3D conformation the protein will take. When a cell synthesizes a polypeptide, generally that chain will fold spontaneously, assuming the functional conformation for that protein. And the folding is driven and reinforced by the formation of different bonds between various parts of the chain, which in turn depends on that amino acid sequence. So amino acid sequence determines protein's conformation or structure, which determines how it works. So there are 4 levels of protein structure. Primary, secondary, tertiary, and then quaternary. Quaternary doesn't exist in all proteins. Some proteins that only comprise one polypeptide polymer don't have quaternary structure. Primary structure refers to the unique sequence of amino acids. And again, that sequence is determined by inherited genetic information. It's not some random linkage. It's not random amino acids thrown together in the hopes that something functional will be created. There's a rhyme and reason to it. Secondary structure refers to the coils or folds in segments of the polypeptide chain, and that contributes to its overall 2D conformation. The coils and folds are results of hydrogen bonds between repeating constituents of the polypeptide backbone. So this is not referring to bonds that are occurring between your side chains. These are bonds between the actual components of that polypeptide backbone. So components of the amino group and the carboxyl group. What happens is that the oxygen and nitrogen atoms of the backbone are electronegative, right? So they have partial negative charges. And the weakly positive hydrogen atoms that are attached to the adjacent electronegative atom have a weak affinity for that other electronegative atom. So they will hydrogen bond. So the hydrogen on nitrogen will hydrogen bond with the oxygen atom of the carboxylic acid on the adjacent peptide backbone. So what this creates is a particular shape. And there's two types of shapes when we're talking about secondary structure. There's the alpha helix, which is kind of this right here. It's this delicate coil that's held together. I think it's held together more specifically by hydrogen bonding between every fourth amino acid. Correct me if I'm wrong on what amino acid it's occurring between. And then the other type is known as the beta pleated sheet. This is when you have polypeptide chains lying side by side, and two or more regions of those chains are connected by hydrogen bonds between those parallel backbones. And that creates a really incredibly strong structure. This is actually what makes spider silk fiber so strong, so much stronger than actually a steel strand of the same weight, which is a very, very, very tiny piece of steel, but nonetheless, it's what allows spider silk to be so strong. So that's secondary structure. The internal bonding or hydrogen bonding of parallel or adjacent regions of the polypeptide backbone. Tertiary structure refers to the actual 3D conformation of the protein. Instead of involving interactions between the backbone constituents, the tertiary structure is shaped or refers to the overall shape of a polypeptide based on interactions between the side chains, so your R groups. And that's all based on the chemical character or chemical reactivity of those different side groups and which ones are adjacent to another and how closely they lie. Some of them will have hydrophobic interactions, others will align closely to form the van der Waals interactions or your London forces. If amino acids that are lying closely situated to one another, if they both have sulfide functional groups, or sulfhydryl, functional groups on their side chains, they will form disulfide bridges. Those disulfide bridges are a type of covalent bond that greatly reinforces kind of the 3D structure conformation. So all of those interactions between the amino acid side chains are what contribute to the 3D structure or tertiary structure of a protein. Now, quaternary structure is the overall protein structure that results from aggregation of two or more polypeptide subunits. So again, not all proteins are comprised of just one polypeptide that has been folded in a specific way. Rather, they are groups of interacting polypeptides. That's quaternary structure. So as I said, Proteins have a variety, or are able to take on a variety of different structures, and therefore have a variety of functions. There are kind of 8 major functions or types of proteins. There are enzymatic proteins, which allow for kind of the selective occurrence or acceleration of chemical reactions. Enzymatic proteins will regulate metabolism by acting as catalysts. And again, that allows them to kind of selectively speed up chemical reactions. There are structural proteins. Structural proteins are for support. Example would be the silk of spider webs, or collagen, which is found in animal connective tissues. There are storage proteins. Storage proteins allow for the storage of amino acids. So ovalbumin, this is the protein that's in egg whites, and that's what provides a source of amino acids for the developing embryo. Transport proteins, these are proteins that enable the transport or facilitate transport of other substances. Example is hemoglobin. Hemoglobin is the iron-containing protein of vertebrate blood, and it transports oxygen from the lungs to other parts of the body. Some proteins are kind of hormonal in function, meaning they coordinate organisms' activities. So insulin. Insulin is a protein hormone that's secreted by the pancreas to help in regulating the concentration of sugar or glucose that's in the blood. The sixth group or sixth type of proteins are receptor proteins. These types of proteins enable response of the cell to chemical stimuli. Right? There's receptors in nerve cells. There are receptors in the plasma membrane that will bind ligands that are used for communication, like intracellular communication. Some proteins provide an element of movement or a function of movement because they have contractile or motive capabilities. Actin and myosin are examples. These are proteins that are responsible for the contraction of muscles. And then lastly are defensive proteins. These are what enable protection against diseases. So your immunoglobulins or antibodies, these are protective or defensive proteins. All right, so changes to protein structure can have a variety of consequences, a lot of them being deleterious in nature. A great example is just a simple change in the amino acid at one placement in the gene coding for hemoglobin, or I should say, one nucleotide substitution that causes the change in the amino acid that's coded for by the gene for hemoglobin can have a huge consequence. What I'm kind of alluding to is the genetic disorder sickle cell anemia. So sickle cell anemia occurs when there's substitution of valine for the normal amino acid glutamic acid at amino acid 6 in the primary structure of hemoglobin, and that causes sickle cell disease, the actual change in the chemical character of hemoglobin so that it becomes sickled in shape and very sticky, which causes these kind of clusters of sickle-shaped hemoglobins, which can clog blood vessels and also pierce vessels, causing an anemic crisis. So it's an inherited blood disorder. But that is just one great example. One small change in amino acid structure can change the chemical reactivity of that amino acid and therefore influence the folding of the protein, which influences function. Protein conformation also depends on environmental influences. So factors such as pH, salinity, chemical conditions, temperature, if those aspects of the environment are altered, that can affect the shape of the protein. In a lot of those cases, you'll actually get what's known as protein denaturation, which is the unraveling of the protein and loss of its native conformation. Oftentimes, once protein is denatured, it becomes inactive. It's no longer a functional protein, which is definitely an issue, because now it's no longer performing a vital function that it previously served in the body. Usually denaturation occurs if you transfer proteins from an aqueous environment to an organic solvent like ether or chloroform. It'll actually, the polypeptide chain will refold so that its hydrophobic regions are now facing outward toward the solvent. Heat is another factor. So when proteins are heated to a high enough degree, the heat will agitate the polypeptide chain enough to actually overpower the weak interactions that stabilize its conformation. And that's why people get worried with extremely high fevers. These are fevers that are above 103 degrees Fahrenheit. At those high temperatures, proteins in the blood can denature, and that's fatal to an individual. Now, usually, once protein is denatured, it cannot be renatured, meaning it will not regain its form. There are some instances where you can have the renaturation after a protein's been denatured, but it has to be dissolved for a specific amount of time and then restored back to pristine environmental conditions that allow for its normal functioning. So usually it doesn't occur, but sometimes it can renature if that denaturing agent's removed. So chaperone proteins, these are proteins that assist with the proper folding of other proteins by creating an ideal environment for spontaneous folding. They work by keeping the new polypeptide or polypeptide subunits segregated from the bad influences of the cytoplasmic environment. And again, that enables it to properly fold spontaneously. So what happens is a polypeptide will, that's unfolded, will enter the kind of your cylindrical chaperone from one end at the detached cap, and then the cap attaches, which causes the cylinder to change shape in such a way that it creates a hydrophilic environment for folding of that polypeptide. And then once it's gone through folding, the cap will come off, and the properly folded protein is released. Alrighty, so those are proteins. Now, the primary structure of polypeptides depends on, or I should say, the conformation of a protein depends on the primary structure of polypeptides. But what determines the primary structure, or the sequence of amino acids making up that polypeptide? Amino acids, the amino acid sequence of a polypeptide, as I said before, is not random. It's programmed by inherited genetic information, and it's more specifically programmed by a unit of inheritance called a gene. Genes consist of DNA, which is kind of provides the genetic instructions for building proteins. The reason why we're talking about DNA is because it's a polymer that belongs to a group of compounds or macromolecules called nucleic acids. Nucleic acids have two types of polymers. You have deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. DNA is the genetic material that organisms inherit from their parents. And again, this is, you can think of the genetic blueprint for the instructions for building proteins. So DNA provides the instructions for building proteins, and proteins are the molecular hardware of a cell that performs different operations or different functions. RNA is similar to DNA, but it's usually single-stranded, has a different structure to its pentose sugar, which we'll talk about in a moment. But ribose or RNA serves as the kind of intermediate that is used during protein synthesis. So this is what actually conveys instructions in eukaryotes from the nucleus to the cytoplasm. And it's the strand of RNA that's read during protein synthesis by the ribosome. So that's what actually is read to create the polypeptide. But it carries information that is gained from DNA. You can think of the whole process of protein synthesis kind of like copying a recipe that your friend can use to make a cake, right? So DNA is the original recipe. When your friend wants a recipe, you don't give them that version of the recipe that you have. You make a copy of it because you don't want to lose your copy. You want to be able to make that same cake. You also can't rely on your friend to not lose those instructions. So you don't want to lose your only copy. So you make a copy of your original instructions. The original instructions are DNA. The copy is RNA, or more specifically, the mRNA strand that is formed or synthesized from a DNA template. Your friend will then use that copy, RNA, to make a cake. Cake represents protein. So that's kind of my dubbed down understanding or relay of transcription and translation, or how DNA and RNA are used to in the synthesis of proteins. We'll get more into the actual nitty-gritties of transcription and translation when we talk about or when we go into our module on genetics. But for now, we're just talking about your basic nucleic acids. So again, DNA and RNA are the polymers of nucleic acids. Their monomers are repeating subunits called nucleotides. Nucleotides are composed of three components. They have a nitrogenous base that is bonded to the 1' or the first carbon of a pentose sugar, and on the 5th carbon or 5' carbon, there's a phosphate group. These 3 components make up your nucleotide. Now, the pentose sugar differs depending on whether it's deoxyribose or ribose. So in DNA, you have deoxyribose as the pentose sugar. In RNA, you have ribose as the pentose sugar. And the two differ based on whether hydroxyl group occurs at the 2' carbon. In deoxyribose, so the pentose sugar that is found in DNA, there is no hydroxyl group. There's only 2 hydrogens that are attached to this 2' or the 2nd carbon. In ribose, so the ribose pentose sugar has a hydroxyl group at that 2' carbon. The presence of the hydroxyl group gives these sugars different characteristics. So because deoxyribose lacks a hydroxyl group here, it's actually more stable in structure, which is why it's able to oftentimes form double strands. It's also the reasoning why we think eukaryotic cells, or most eukaryotic cells, went on to have double-stranded DNA serve as the primary genetic material in their cells. Now, going back to the nitrogenous bases on nucleotides, there are 2 types or 2 categories of nitrogenous bases. You have pyrimidines and purines. Pyrimidines are 6-sided rings, and there's 3 of them, or 3 kinds. You have cytosine, thymine, and uracil. Now, thymine is only found in DNA. Uracil is only found in RNA. Purines are a little more complex in that they contain the same 6-ring structure, but they also have a 5-ring structure fused to it. So they're kind of double ring structures. And there's 2 of them, adenine and guanine. Both of these are present in DNA and RNA. Now, nucleotides are linked together to form these polymer chains, which represent DNA or RNA. And they're linked by what's known as a phosphodiester bond. And that bond forms between the phosphate group of one nucleotide and the 3' hydroxyl group on the sugar of the adjacent. So it's forming between one of these oxygens on the phosphate group and between the, let's get to a good picture here, between your hydroxyl group on the 3' carbon. So they're linked together in that fashion by phosphodiester bonds to create chains of nucleotides that usually have a 3' to 5' or a 5' to 3' orientation. That orientation is just referring to which end is kind of freed at the end of the polymer. So at one end you'll have an exposed 3' hydroxyl group. At the other, you'll have the phosphate group on the 5' carbon. So we'll get more into this with talking about genetics. But you have the phosphodiester bonds occurring along the backbone or the sugar phosphate backbones of your nucleic acid. And then the nitrogenous base pairs or bases will base pair together via hydrogen bonds. Thymine hydrogen bonds with adenine or base pairs with adenine, guanine base pairs with cytosine. So this is for DNA. In RNA, you have uracil instead of thymine. Alrighty, so that's the important stuff for your proteins and nucleic acids. If you want additional information, you can post in the comment section, or if you're one of my students, ask me in class or during my office hours. Again, there will be more information about proteins and nucleic acids when we talk about genetics, or you can refer to the lectures that are already posted from the previous genetics classes that I have taught. I hope this lecture provided some useful information for you and that you have a better understanding of macromolecules than you did before.